Hello and welcome to the meeting of the Rotary E-Club of District 7710 for Tuesday, April 10th, 2018, and thank you for joining us. I just want to give you a few quick reminders about the ways you can connect with our club, along with attending the regular meetings that we post on Facebook on Tuesdays, and in the newsletter. You can reach us and find out more information at the website Lead With Us, that's leadwith.us. And if you visit the page, you'll be able to learn more about the club and contact us by email link. We're also, as I said, on Facebook, and if you look for Rotary E-Club of District 7710, you'll find us on Twitter at Lead With Us and Instagram at Lead With Rotary. So please take a moment to follow or friend us. So tonight it's my absolute pleasure to introduce my friend, Caitlyn Cooper, who I met because she, the first week of graduate school at the Nicholas School, she applied to work with me, and I was so excited because nobody ever wants to work with me. my work tends to be a little boring, and Caitlyn came with all the enthusiasm that I think I've ever seen with anyone come to an administrative position, and she also brought an immense amount of skill, and I actually probably learned more from her than she learned from me. So I'm really excited that she's here to talk with us this evening about her lemur research, and I've asked her to share some other tidbits of information that are pretty cool. So I'm going to actually just give you the full Caitlyn introduction. I wanted to let you know how I knew her. So Caitlyn, as you probably can imagine, has always loved wildlife, but after learning about endangered lemurs in her high school zoology class, her interest was piqued. Lemurs then stayed at the back of her mind for the next 6 years, and she went on to study at Indiana University. Caitlyn originally started out as a biology major and then realized she was much more interested in the big picture of biology than she was of the microscopic proteins or processes. Can't imagine why. Her sister recommended that she check out the bachelor's degree in environmental science instead, so she switched to this track and was much happier taking classes that looked at the environment as a whole. So when she decided, and she was thinking about going to graduate school, her advisor said to search Google Scholar for research topics that she was interested in and to see which universities the authors had come from. And after a quick search of lemur conservation, her path led to Duke, and every single hit on the Google search also led to Duke due to the Lemur Center being located here in Durham. So she moved to Durham 2 years ago to pursue her master's in environmental management at the Nicholas School of the Environment. Her time at Duke has been filled with new experiences and learning new skills. She was co-editor of Eno Magazine, an environmentally inspired arts and literature magazine. The publication celebrates the environment through artistic submissions from people both in and outside the Duke community, and she enjoys all forms of artistic expression. So Eno has been a great creative outlet for her. So with graduation fast approaching, she's looking for positions, and she did just tell us that she has an interview this week. So she's looking for positions at environmental consulting companies that allow her to conduct wildlife and habitat surveys on endangered or threatened species. And after 11 years of summer camp in the wilderness of southern Tennessee, catching salamanders, exploring creeks, and tromping through the woods, was never something she quite grew out of. So if anything, Caitlyn found a way to make those childhood adventures a career. And I'm so happy for you, Caitlyn, and excited to have you here to talk to our club tonight. So thank you for being here. And I will let you unmute and take it away.
All right, well, thank you. I hope you guys, if you have questions, I would love to hear questions because I presented at the Duke Lemur Center. And surprisingly, they didn't have any challenging questions. And then at my master's project presentation, I still didn't have many challenging questions. So feel free to challenge me. I actually enjoy getting to talk about this since I spent so much of my time on it. So I will get right into it. So I studied lemur species habitat relationships at multiple spatial scales in Ranomafana National Park. And it was a little difficult to find a research project. So, by the way, on your screen, Eve, I see, so on the right-hand side, I see the video, but is any of your screen blocked, or do you only see the slides? Are you missing out on the pictures?
No, I think we can see the picture.
Okay.
I see.
I just don't know how it turns up on everyone else's screen.
Yeah, I think it's all there.
Okay. So basically, I knew I came to Duke to study lemurs. That's why I'm here. And that's not necessarily the track I'm going to be on for the rest of my life. Obviously, I'm looking for positions on other endangered and threatened species, but I knew that this was something I had to pursue and I had to do, and I wasn't going to be happy unless I did it. So I knew I was coming to Duke to study lemurs, regardless of whether or not it was feasible, but I made it feasible. So I talked through these project ideas with my advisors, with friends, with other primatologists. We really looked at what limitations there were. So I was very limited by time. So conducting this study in one summer, 3 months, is relatively short. Cost was a huge issue. Doing international work is always incredibly costly. And then feasibility, which the feasibility I can't say I found out until I got there and looked at the environment and walked around and actually saw what was realistically possible. So even up until the entire planning process of writing a proposal, writing out my methods, what I planned to do, it still all just went out the window and changed once I got there. So that's always something that happens with field work, is sight unseen, you don't know what's going to happen. So we kept revising it until we came up with an answerable research question and eventually narrowed it down based on what my advisors wanted me to be able to do. So it did not start out very well. So to go to Madagascar, I had to take a malaria prophylaxis. And the first one I was prescribed was doxycycline. Now, doxycycline made me so dehydrated the night before my flight that the morning of my flight, I ended up having my roommate drive me to urgent care to get an IV because I was just not going to give up a $1,700 flight. So I wasn't about to miss that. So then once I got there, I thought I was good. I was eating the food, and I'm an incredibly picky eater, but I was eating the food, and then about 2 weeks in, I got really sick, and I just had stomach issues. So I was saying always bring ciprofloxacin, I think is how it's pronounced, or Cipro for short, because ultimately I needed that to get past a different food environment. I spent a lot of time waiting on permits, which was really frustrating because they were supposed to be ready for me when I got there, but 2 or 3 weeks in, they weren't ready. There's also a ton of holidays that happened right when I got there, so I had to keep giving off days work. And I felt like I was really too lenient in the beginning because I was having these half days because I was trying to get a feel for the environment and the trail system and what was there and what was possible. And I was trying out these methods, and in the end, when we were working, a 7-day-long field excursion, they were like, "Oh, we don't get the weekend off." I'm like, no, it's a short project. We have to keep working. So I actually hired 2 technicians, and then I had a student with me, a Malagasy student, and the technicians were Malagasy as well. But this is required for the government to come and do research in their country. You are required to work with a student to help them, and they help you. And so if I were to bring 10 undergraduates with me, then I would then have 10 more Malagasy students that would be paired up with each of those. So I didn't have anyone with me. I didn't bring anyone. So it was actually really nice to be able to hire people once I got there that were more familiar with the environment than I was. And then finally, I took this, I switched from doxycycline malaria prophylaxis to Lariam. And I don't know how familiar you are with those sort of drugs, but it has, I'm going to go to the next one. It has a lot of side effects that have to do with depressed mood and having crazy nightmares and dreams. And I basically cried for 2 weeks straight, every day, because Lariam was just screwing with my head and my emotions. And that was awful. So it was hard to get past the food and the stomach issues and the malaria prophylaxis. And in the end, we finally got into the field and got started. So you might be wondering, why study lemurs. Why am I so fascinated by them? Well, I remember finding out about lemurs and how unique of an animal this was. And then shortly after finding out they existed, I found out they were all at some level of risk. So from threatened to vulnerable or endangered or critically endangered. And most of this is caused by habitat loss and fragmentation. And in my mind, I saw that as something that's preventable. It's an anthropogenic process. It's something that is caused by humans that could be managed better to not cause all of this extinction. All lemurs are endemic to Madagascar, so they're not found anywhere else in the world, which I just think, again, that's incredible how unique this island is and how much diversity is there. It's a huge biodiversity hotspot, not only for lemurs, but for plants and other wildlife. And it's the 10th forest nation in the world. So most of this habitat loss and fragmentation is coming from something called TAVI, which is slash and burn agriculture, and basically clearing land to create agricultural crops to feed their ever-growing population. So I studied in Ranomafana National Park, and this is in the southeastern side of Madagascar, and this is a tropical rainforest, whereas the western, southern sides are more desert-like, more spiny forest and a lot drier. And then the park itself was established in 1991, and it was preserved from exploitation because of how mountainous it was, which that kind of gives you an idea of how many mountains I had to hike this summer. If most of the people there didn't want to hike it, I guess I did. But so you might be wondering, why would I have chosen this park in particular? Because I could have gone anywhere in Madagascar. And first of all, I was invited to come by Patricia Wright, who is one of the top lemur conservationists in the world. And she created this research station to facilitate research in Madagascar. Basically, it made it a lot easier. There's a place to sleep, there's dorms. It allows you to collaborate with other primatologists and even just other researchers who are studying bats or moths or who knows what else. And they have Wi-Fi, which made it really easy to stay connected with my family because, of course, your parents go crazy when you go out of the country and they worry about you, so you can stay in touch. And then I was able to stay in contact with my advisors when things were going not as I had planned and talk about alternate options. But aside from there being a field station there, they have a unique species that isn't found anywhere else. They have highly threatened species, and they have multiple levels of disturbance, which I'll get into a little bit later. So hopefully you enjoy all the pictures. I tried to put in as many pictures as I could because they're cute. And I actually took all of these except for the greater bamboo lemur because, as you can see, the Ranomafana National Park is home to 7 lemurs that are diurnal, so during the daytime lemurs, and they're all some level of threatened. And then specifically the golden bamboo lemur is only found in Ranomafana National Park. And this is an incredible lemur because they are the reason the park was established. They found this new species and then realized it was nowhere else, and so they created the park around this lemur. And then the greater bamboo lemur, it is elsewhere in Madagascar, but there's only 2 individuals left in Ranomafana National Park. So it will most likely go locally extinct. And I can tell you more about that if you're interested in it, because that was one of the questions someone had asked me, was why there was only 2 left. So there has been a lot of previous research in this park, mainly because that research station is there. It really promotes bringing people in and encourages people to study there just by having, you know, kind of, I guess you would call it an American hub where you can find other people who know the same language as you, and you can really collaborate with other people who know what you're studying. Now, Johnson et al. In 2005, or he published this in 2005, but did this in 2002 and 2003, shortly after the park was established. And he did this huge biodiversity study on everything. It included human disturbance and diurnal animals, nocturnal animals, birds, insects, chameleons, everything you can think of. And what's interesting about that was in the southern section, he found that the black and white ruffed lemur, which is normally only found in really pristine sites, was actually found in higher abundance in the south, which is considered a highly pristine site. So it hasn't been logged or disturbed by people because it's pretty far away from any villages. And then Ganshorn found that low intensity logging actually led to more lemurs. So you normally think that logging would be a bad thing, but at low levels, he found that it led to more sunlight, which increased the protein content of the leaves, which is really good for folivorous species. And then it also increased fruit production, which is really good for frugivorous species. And the pre-logged state, he didn't find any significance with that, but the low intensity logging increased the number of species he found and the abundance, but in intensely logged areas, it was actually negatively associated. So that was not a place where lemurs were found. To further complicate this, so we've seen the black and white ruffed lemur in pristine sites. We've seen a few species in minimally logged sites. And then James Herrera studied something similar in 2 locations of Ranomafana National Park. And the 2 locations highlighted on this map are the same ones I went to. And he found that not only did 3 species have a positive association with the minimally logged site, just like Ganshorn found, they also found 1 species in the heavily logged site, which you wouldn't expect. So there's something going on here where all these species require different microhabitat factors that are going to affect their distribution and abundance differently because they all have different diets. And even the 3 bamboo lemurs, who all 3 eat bamboo, are specialized to eat 3 different parts of the bamboo plant. So there's clearly a lot of interactions going on here that aren't well understood. And often when people study lemurs, they either study, sorry, my cat's in the background, they either study their behavior or they study 1 species. And I, like I said, I really like that big picture. I want to see what's happening with as many species as I can and their entire habitat. So to show you how I think this project is unique from others that have been done, the multiple spatial scales is basically, the microhabitat is what's in 1 small 20 meter by 20 meter plot. And then the landscape scale, the large scale, is like distance to road, distance to village, distance to things that are a lot further away. And then I also did these point plots. So I just made a random map of points and then put a vegetation plot on top of it. And that was where I studied. Most people who study lemurs and their abundance or their composition, they do it on a transect. And a transect is usually in a straight line. And that requires actually cutting the vegetation in the park along that entire route so that you can walk the route over and over and over again, upwards of 30 to 50 times. Or they'll do it on an already made trail, so like a circuitous route on a trail, which isn't necessarily, I feel like it doesn't really talk about their habitat per se. So I wanted to try a different method that hasn't been tried before. I included a ton of habitat variables because I really wanted to just look at everything. And then I did some really in-depth measurements, which again, I'll talk about later. So I think this is really important because it helps us continue to monitor a recovering forest, which hasn't been done really extensively since 2008. And it helps us also see how lemur composition is changing as the forest is actually recovering, because we can't expect it to be static. It's always changing. And therefore, I think lemur composition is also going to change as well. And by doing multiple spatial scales, I'm allowed to do a more robust analysis and able to hold different variables constant to see what causes what more clearly. So this northern section of my study area, where the brown dots and then the yellow star is actually the research station, Centre Val Bio, and then really close to that is the entrance to the park. So the northern section is called Tala Tekeli, and I'll just refer to it as Tala. And this was the heavily disturbed site, and it was heavily logged in the 1980s. And it is the most accessible to tourists, and there are tourists in there constantly because it's so close. And then you'll see the southern section is Vatu, Haranana, and Valahoka, which I split into in this map here showing Vatu and Valu. It's easier to just say the shorthand of the name. And those are a lot further from the entrance. So Vatu is technically visited by tourists, but it's not daily. Maybe they do a 2-hour hike out there and camp for the night, but it's not heavily visited. And then Valu is 3-hour hike away, and I mean, you have to bring all your stuff, so it's not an easy trip to make, especially on how mountainous it is. So that's only a researcher site. So there shouldn't technically be any disturbance out there, because I hope that researchers are careful to not cause any disturbance. Now, Vatu was logged of 1,000 trees in the 1980s as well, plus however many trees were cut down on the logging road to make a road to get the trees out. And then Valu has never been logged and is very minimally visited by anybody. So my first question was looking at, is there a significant difference in these habitat characteristics between these 3 sites? And then my second was, is there a significant difference in lemur composition or occurrence, so either their biodiversity or abundance between the 3 sites? And then finally, I wanted to see if there was a significant relationship between lemurs and particular habitat factors, which we'll go through here. So my lemur variables was abundance, species or biodiversity, and then number of observations. So I recorded the number of times that I returned to a plot to look for lemurs. And then I had a ton of microhabitat variables. So you don't have to look at all of them, but there are 2 main ones I wanted to highlight, and that is the fruiting trees, which is a food source for several of our frugivorous lemurs, and then the bamboo stems, which I included because they're the 3 bamboo lemurs. Now, there are 3 types of bamboo in this area, and only 2 of them are measurable because they're big bamboo stalks, whereas the other one is a vine that just kind of hangs low, and you can't really count that or measure it. And then my landscape analysis also included anthropogenic factors because I'm really interested in this human impact element. But the good news is this park is really protected, so there wasn't actually that much impact. I didn't see any lemur traps or any sort of exploitation going on. I did find 3 or 4 sites where trees were cut and exploited for honey, but none of those were in my plot, so I couldn't really do that analysis. But then I looked at big scale things such as elevation, distance to the forest edge to see how far the edge of their habitat is and impacts their presence, and then distance to things like the main trail or our camp, since we can't deny that we as researchers being there to some degree must be making an impact. And then finally, we had measured latitude and longitude and analyzed that as well. So my plots were 20 meters by 20 meters, and I had a team botanist who told us the tree species in each plot. We estimated tree height and canopy width, and then I measured the tree's diameter at breast height. And then we also measured the amount of canopy cover in each plot and estimated herbaceous ground cover with this figure, which is basically you kind of hold it out in front of you. I had it in a Ziploc bag at each plot and kind of looked at it and looked at the ground and then took an estimate against all 4 because it's broken up into 4 subplots, I guess. So I took that at each 4 and averaged it, and then just did my best to estimate. It's not a very strong measure, but it's a good estimator. When we had me and the 2 technicians and the student, we were in a group of 4, and we slowly crossed the microhabitat plot looking for lemurs. So after we measured all these habitat variables, we would come back the next day to look for lemurs, and then the next day for lemurs, and we would keep returning until we ran out of time. And so we would cross the plot for 10 minutes and going quietly, taking pauses to observe our surroundings. And when we found a lemur, we paused the timer, took data on that species, and then went back and continued to cover the plot and restarted that timer. Towards the end of the field season, we wanted to get a bigger sample. So we split into 2 groups and to cover more land. And instead of going 4 across, we took a slightly circuitous route and came back down. And then instead of looking for lemurs for 10 minutes, we did it for 20 minutes so that we would have an equal sampling effort across plots. So I returned to each plot to look for lemurs 6 to 9 times, depending on where it was and how long the field expedition was. And then we took GPS data of every lemur we found. And not only did we do this within my research plots, I also took data opportunistically, which means I have hundreds of GPS points of where we've seen lemurs, which we've given to the park and they can use as they wish. I wasn't able to actually analyze the opportunistic data, but I still think it's good to have and to measure which trees they're in, or if they have any infants with them, males and females, adults and juveniles, and that sort of thing. It's really interesting to look at the composition of lemur communities in this park. And then finally, I analyzed my landscape variables using ArcGIS by just measuring the distance between my plot and things like the forest edge or the camp. Now, for my statistics, I did run Mantell's test, and you don't have to know anything about Mantell's except for the fact that it just compares matrices of data and looks for differences. And it accounts for covariance, which means it's basically a non-parametric test that allows me a lot more freedom, and it accounts for variability in the data. So it can answer questions like, are sites that are dissimilar in lemur species composition also dissimilar in canopy cover after accounting for all the other habitat variables? And I did this for lemur species composition and abundance, and then for lemur species presence absence. And the presence absence data gives species who are present a 1 and absent a 0. So all species have equal weight regardless of how many of them were there. So 2 different tests look at 2 different things. And then I did a ANOVA analysis of variance against these 3 sites. And I wanted to see if their habitats were significantly different and if lemurs were significantly different across the 3 habitats. So to answer how much stats did I actually do, it was a lot. It was 700 something statistical tests. Most of those were Mantell's, and I did all of this in our statistical software. And so I'm glad I learned that skill, but it was stressful. It was a lot of being inside at the computer instead of outside in the forest. So, but it's part of the job. And then when I was looking at these habitat variables across the site, so are these habitats different? I found several different variables that actually were different across the sites. And to just remind you which site's which, so the Tala, the heavily disturbed site, was really different in microhabitat from Batu, the minimally logged site. And for some reason, Batu and Balu, the minimally logged site and the pristine site, there wasn't significant difference between these variables. So it's kind of hard to say what it means, but as a general inference from this, the 3 sites are different and across these 3 locations. So, I mean, I can't necessarily speak to why one variable is significant in one category but not in another, but it just goes to show that there are differences occurring. Now then, when I was looking at if lemurs were different across sites, not only did I look at each individual lemur species, I looked at lemur diversity as a whole and overall lemur abundance. And I did not find any significance. I couldn't even calculate this for the greater bamboo lemur because, like I said, there's only 2 individuals left and none of them showed up in my plot. They were pretty stationary in one patch of bamboo that wasn't in my study area. So that was kind of interesting to see that there wasn't any differences. And then when I was running my Mantel's test, I originally grouped all my habitat variables together in these like mega matrices to just look at overall trends. And there was no significance in that with lemur species composition or with the presence-absence data. So essentially, when you look at them as a whole, they're not significantly different from each other, if that makes sense. But then when we looked at individual species and these habitat variables, we did find significance for the red-bellied lemur, the golden bamboo lemur, and the black and white ruffed lemur. Now, as you can see, there's more in the presence-absence column than there is in the species composition column. You might be wondering why my one variable show up in one column but not the other. And essentially what this means is, so a high tree density or low tree density may be necessary for a species to be present, but no amount of changing that density is going to influence their abundance. So that was an interesting thing to see, that the black and white ruffed lemur had a lot of variables that were important to them for them to be present, but not necessarily to change in abundance. Now, the red-bellied lemur was negatively significant with distance to camp, which means they would be further from camps. And I thought that was really interesting because out of all the lemurs that I encountered, they were the most friendly and they would come down the tree to people. But it's true, I never saw them at a camp. So that was interesting. And then, as we would expect, we found significance between the golden bamboo lemur and bamboo stalks, their food source. And then when I was answering these questions, yes, I found that these habitats are significantly different in the microhabitat and the landscape across all three levels. And then when I was answering the second question, there wasn't any difference in lemurs across the three sites. And I thought that was really interesting because that was completely different from what James Herrera studied, from what Ganshorn found, and what from the Johnson study in 2005. So I didn't find what everyone else was finding. So that was a really interesting insight. And then when I was looking at these individual habitat variables in comparison to specific lemur species, it's these specialist lemurs that really are driving, or that are really, how do I phrase this? That really needs special habitat factors, basically. So the red-bellied lemur is a frugivorous lemur, a strict frugivore, and then the black and white ruffed lemur on the left is also a strict frugivore, and then that golden bamboo lemur on the right, most of its diet, like 90% of its diet, is strictly bamboo. So it seems like these specialist species have really important habitat requirements. And then I really wanted to highlight the black and white ruffed lemur because we found significance between the black and white ruffed lemur and canopy cover, which James Herrera also found in 2008. So it goes to show that doing this with a different methodology is still showing similar results, even though it's not showing that similarity across sites. So I know that was a lot all at once, and I think I was talking really fast, so I apologize. Hopefully that'll leave time for questions. But you might be wondering, so why can't we detect differences in lemurs across these sites? Is it possible that the forest recovered after only 30, 40 years? That's not incredibly likely, but it's possible. Or is it just that some variables aren't important? So when you compare the habitat factors that are different between sites and the habitat factors that are significant for lemurs, there's not a lot of overlap here. And essentially what that means is if a variable is important, significantly important for a lemur to be present, but those variables aren't different across the sites, then it's not really going to influence their abundance across those sites either, or their presence. The one exception to this was I did have canopy cover in both columns, and it could be that it's important for black and white ruffed lemurs, but it's not as important as something else like diet or following their mates, because this is a female phylopatry system, which means the woman is in charge. There's one dominant female, and everybody just follows her. So if she just decides to go somewhere else, I mean, regardless of canopy cover, they're going to follow her, whether it's their habitat preference or not. So it's just, I can't answer every question, but that was one that I found really interesting, that it might just not be as important of a factor. So it is possible the forest has recovered, but considering forests typically take upwards of 60 to 80 years to recover, I really don't think that's the case, especially since we saw differences in those habitat factors. So it's more likely that the habitat variables that are important for certain species just aren't different across all three sites. So some of the limitations of this study was GIS related. So I didn't actually get my GIS data until about halfway through the summer. And the bounds of Vatu and Valu are really fluid. It's so hard to tell where one ends and the other begins, and where this logging occurred because it was so little. And the first graph on the left shows James Herrera's study from 2016, where he studied Valu, so the southernmost site. The middle one was mine, showing where I studied both Vatu and Valu, and then the most right was another study where James Herrera studied Tala and Valu, or Vatu. So like where Valu is and where Vatu is really unclear. So it seemed like the technicians knew, they would be like, oh, we're in Vatu, so they had some way of knowing, but it wasn't obvious to me, and I cannot draw a specific map showing this. So I feel like that could have impacted it if I wasn't necessarily putting the data points in the right categories. So if I had a plot that I thought was Vatu and it was actually Valu, or if they're just so similar that at this point it might not matter. And then I was also limited by where I could go on a trail. I originally had this map of the park with just points all over it that I was going to go to. And I realized that is absolutely impossible, completely not possible, based on how mountainous it is and how dense it is. You can't just go to some point on a map and be like, yep, we're headed there. It has to have a trail connecting it, because there's no way you'll ever get there. And so this was really interesting just to see a different method, because nobody's done these point methods that I've done. They usually do these transects, which, like I said, are either on a trail or they cut vegetation to create a trail, to create a transect. So it was interesting to see that this method that hasn't been done before on lemurs, but has been done on other species, actually can work for lemurs. So in the future, I think it would be really interesting to do this at all of the park, and then not including pseudo-replication. So what that means is you almost always, in statistics, you want to have a sample of 30, and some made-up number that somebody said 30 was the right amount. And even though I had 26 plots, which sounds close to that sample of 30, realistically, I only had 3 sites. So it's pseudo-replicated. It would be nice if we could have 10 disturbed sites and 10 minimally disturbed sites and 10 pristine sites to really be able to make these inferences more solid and be able to apply what we found to other parks. Because right now, with a pseudo-replicated study, I can really only apply this knowledge to this park because I haven't studied sites outside the park. I think it would be really interesting in the future if somebody could reevaluate the degree of degradation, because it has changed so much, and maybe really map the park and what they consider primary forest and secondary forest, and mapping that level of impact would be really interesting. And then, but I know that's not realistic because, like I said, half the park isn't even accessible because there's just no trail system. And then I would like to see this more in lemur science, people actually taking temperature and precipitation data and weather data, which has actually been absent from primatological studies lately. And I don't know why. I think it might just be because it's so variable and it's just one more thing they have to measure. But it would be interesting to see if those things are significant because I can guarantee you, if it was pouring down rain, we did not find lemurs. We absolutely could walk around for 5 hours and not see a single one, just because the detectability is so low because they're just cuddled up in a cold ball in the tree trying to wait out the rain. So it would be really interesting to include those weather factors. So far, James Herrera actually did do a study 1 year prior to this that did cover the whole park, again doing transects. And he did include one landscape variable of elevation, but didn't really include any others. And then he did include, oh, there's an echo, habitat disturbance, but he was really only looking at historical habitat disturbance. So like I said, whether or not it had been logged, instead of looking at, okay, how are people affecting it now? Because even though it's a relatively well-protected park, people are still cutting trees down for honey. And so there is still disturbance occurring, and it should be measured and not just looking at what happened 40 years ago. And then in his study, he also looked at phylogeny and phenology, which I thought was really interesting. I also think it would be good to replicate this study on nocturnal species. It's kind of impossible to do both diurnal and nocturnal species in one summer unless I had a whole other team to do the night surveys. But I just think it's really interesting to identify which habitat factors are important for certain species, because if you're trying to find a new protected habitat, or you're trying to find a new patch of land that you could protect, that would be ideal for a critically endangered species that's on the brink of extinction, then it would be good to know which habitat factors are important, to know which areas to prioritize. So if, say, the sportive lemur really needed a high amount of canopy cover, and you have two sites you're deciding between, you don't know how to choose, I would choose the site that has a higher amount of canopy cover because that's essential for them. I would also like to see this study be done on a larger sample size, as I said, having 10 disturbed sites and 10 minimally disturbed sites. I was saying ultimately this could be someone's PhD if they had 5 years to field seasons to do this over and over again. I was very limited in the amount of time that I had. So I just wanted to thank my advisor, Nikki Cagle, and then I had a ton of grants give me funding. And then I had a lot of people at the Nicholas School and the Duke Lemur Center who helped me get through every little thing that went wrong, from illness to my GPS not working to just not knowing how to hike in a mountainous rainforest. So I had a lot of support, and I couldn't have done it without them. So that's it.
Great. Thank you so much, Caitlin. That was amazing having you here tonight.